So I will talk today about another topic, which is the cell cycle modeling, and the talk will be shorter than yesterday because I will not need the introduction which I gave yesterday. So I will try to switch to the slides like that. Like that. Does it work? Do you see the slides?
Yes, we do.
Okay. Okay. So the topic of this second talk is modeling the cell cycle, and I will discuss different cell cycles in different organisms, including amphibian embryos and mammalian cells. So yesterday I showed this table, which presents the main rhythms observed in biological systems according to their period, which is increasing. And I discussed yesterday circadian rhythms, which are rhythms which occur in all eukaryotes and some prokaryotes with a period of about 24 hours. And today I will discuss the mitotic oscillator, which is driving the cell division cycle. And we will see that the period can go from some 10 minutes to 24 hours or more. And at the end, I will show that these major cellular rhythms, in fact, are coupled. And there is crosstalk, as one may say, between the cell cycle and the circadian clock. And we will see what are the dynamical consequences of this and how this can be modeled. So the cell cycle, of course, plays key roles in biology. In developmental biology, it is, of course, needed to go from the fertilized egg. I realize that there is an eye which is missing there. The fertilized egg to the adult organism. But deregulation of the cell cycle can have also profound consequences. For example, the development of tumors leading to cancer. And I will also discuss this aspect in the talk. So using mathematical models can be useful if they are based on experimental observations, because they can throw light on the dynamics of the cell cycle and the different types of dynamical behavior which can be associated with regulation of the biochemical network driving the cell cycle. Experimentally, the cell cycle has been studied first in model organisms, besides, of course, bacteria. Yeast, for example, has served and continues to serve as a very useful organism to study the cell cycle because there are many mutants which are available. And many mutants of the cell cycle, which are called CDC mutants, cell division cycle mutants, were characterized in yeast and then studied in other organisms, including the amphibian egg. Xenopus laevis is another organism of choice in which many advances have been made to understand the biochemistry of the cell cycle machinery. There are two types of cell cycle which can be distinguished. In the frog, the 12 early embryonic cell cycles are relatively simple. When I say sometimes they are simple, then people who work in this field, of course, are very annoyed because, of course, it's not simple. It's rather complex. But in comparison to other cell cycles, like in mammals, it is indeed, it has a simpler structure. The first 12 embryonic cell cycles are rapid, with a period of about 30 minutes in the frog. In mammals, typically, one distinguishes four phases: the G1 phase, S phase, G2, and M. S is the DNA replication phase, M is mitosis, cell division itself, and S and M are separated by these two gap phases, the G1 and G2 phases. And typically, this type of cell cycle, which is also observed in yeast, by the way, with the four phases, can go from some 8 hours in yeast to longer times, like 16 hours, 24 hours or more in mammalian cells. So I will start with the frog, where the first division cycles are rapid, as I said, 30 minutes, about 30 minutes. And so the question as to what is the biochemical machinery which is driving these cell cycles has been studied much in the 70s and the 80s. And the view which was reached at that time was that these early cell cycles in the frog embryo consist in sequences of an interphase and an M phase, mitosis itself. And the mitosis was found to be triggered by a factor, a protein factor, which was called MPF. MPF for mitosis promoting factor or maturation promoting factor. So there was a peak of MPF bringing about mitosis, and then an interphase leading to the next peak of MPF. And once mitosis was completed, MPF was inactivated. So I'm really reviewing the salient features of the phenomenon for those of you who were not exposed to the topic before. And it was also found that MPF is a complex between two proteins. One of the proteins is an enzyme, which was found to be CDC2. So CDC2, it's a cell division cycle mutant, which was characterized in yeast. And CDC2 is a protein kinase. So kinase, an enzyme that attaches a phosphate group to another protein, and thereby can activate it or inhibit it. In this case, this type of phosphorylation inhibits. That's another kinase. No, CDC2 is a kinase which phosphorylates substrates and which eventually leads to cell division. The other protein making the MPF complex was found to be cyclin. Now, cyclin is a member of a family which was discovered by Tim Hunt, who asked the question if the MPF is activated periodically, is there a protein which varies cyclically in the cell cycle? And he characterized one of these proteins, and he called it cyclin. And so MPF is a complex between CDC2, the kinase CDC2, and a cyclin. And what Andrew Murray and Mark Kirshner, who were at Harvard, showed in 1989, a beautiful paper, is that cyclin synthesis is driving the early embryonic cell cycles in frog embryos. So this is really what is driving the succession of the early cell cycles. And the scheme, which is shown on the screen, shows that there is an oscillation. Cyclin is building up to a threshold, triggers the activation of MPF, and then this brings about mitosis. And at the same time, MPF is inactivated and cyclin goes down. So there is an oscillatory pattern of cyclin synthesis and degradation and a pattern of periodic activation of MPF. And the question is, what is the mechanism for that? So as you may know, the Nobel Prize in Physiology or Medicine in 2001 was awarded to 3 scientists. I already mentioned Tim Hunt. Also, there was Paul Nurse, who characterized the kinase CDC2 in yeast, and Lee Hartwell, who characterized a number of mutants of the cell cycle in yeast. And so they were awarded the Nobel Prize in Physiology of Medicine for the discoveries of key regulators of the cell cycle. This is a figure taken from Paul Nurse's work, where you see that he measured the activity of the kinase CDC2. And so it's not possible to measure that easily for many cycles. And here is, of course, an important difference with respect to modeling. You will see modeling these oscillations of CDC2, and a model, of course, can produce readily many cycles, as many as you wish actually, of one of the oscillating variables. Experimentally, it's much more delicate and difficult to do, but this is one of the cases where at least 2 peaks of CDC2 have been shown, and this is the key issue. Once you show that after a peak, spontaneously the system can produce the second peak, then then you have shown all the ingredients which are needed to explain sustained oscillations. Mitosis is triggered, that's the other curve, is triggered after the increase of the kinase CDC2. Now, what about modeling these oscillations? In 1991, so it was already a long time ago, John Tyson and I, actually independently, proposed a model for these oscillations in CDC2 and cyclin in frog embryos. And John Tyson, whom I know, of course, very well for a very long time, based his model on positive feedback, while I, as you will see, focused on negative feedback. The model I proposed is called the minimal cascade model for the mitotic oscillator involving cyclin and CDC2 kinase. So the reason why I came to build this model was when I read this paper published in Nature in 1990 by the group of Eric Carcenti, who works at the EMBL in Heidelberg. And you see that Tim Hunt was one of the co-authors of that paper, and Marianne Felix, who is now a developmental biologist working in Paris. So what they found, and what they showed experimentally, is that when you add 2 extracts containing active MPF, active CDC2 kinase, when you add CDC2 kinase, it triggers cyclin degradation. So what they suggested, so this is a sort of negative feedback, because cyclin is needed to activate CDC2 kinase, and once CDC2 kinase is activated, it destroys cyclin. So this is negative autoregulation. And what they suggested in this letter to Nature is that this negative feedback could be responsible for oscillations in CDC2 activity, provided there are some thresholds and some time delays. So the question is, once you consider such negative feedback, what is the source of thresholds and of time delays? And so this is the model for the embryonic cell cycle, which I proposed at that time. So why do I come back to that model, which was published so long ago? For 2 reasons. First of all, because I want to give you a kind of progressive account of how models are being proposed. This is a relatively simple model with 3 variables. You will see later in the talk that one can consider much more complex models for the mammalian cell cycle. And also because this is the way the progress was made, to start with modeling the embryonic cell cycle and then going to more complex cell cycles after that. And another reason why it's interesting to consider this system is that it can serve as a building block which will reappear in the complex model I will show for the mammalian cell cycle. So some of the actors are similar, by the way, remain the same. So this model takes into account phosphorylation, dephosphorylation, and negative feedback exerted on cyclin. So cyclin is synthesized constantly at a certain rate, VI, and then degraded. And cyclin accumulation leads to the activation of M, which is CDC2 kinase. The active form of M of the CDC2 kinase is called M, and it can be inactivated by phosphorylation, by a kinase called V1. So M plus is the inactive form, the phosphorylated inactive form of CDC2 kinase. The enzyme, which has a rate of V1, is a phosphatase called CDC25. Now, how do we represent negative feedback in this system? Taking into account the fact that CDC2 kinase phosphorylates proteins. So it was assumed that active CDC2 kinase activates a protease, and this protease now it's called APC, the anaphase promoting complex. So V3 is the rate of phosphorylation of this protease, and the V4 is a phosphatase which inactivates the protease. And what does the protease do? It degrades, it phosphorylates cyclin and degrades cyclin. No, it's not. It does not phosphorylate. It is phosphorylated, but it degrades cyclin as a protease does. So what are the three variables? The three variables are the cyclin concentration, C, the fraction of active CDC2, M, and the fraction of active protease, X. And these equations are not highly nonlinear. They are, the highest nonlinearity is of the Michaelis-Menten type. That is, that is, for example, degradation of cyclin proceeds at a rate C over Kd plus C. That's the Michaelis-Menten expression. However, it is multiplied by Vd, the maximum rate, times X, which is the fraction of active protease. Okay, so these equations are not highly nonlinear, but they are coupled. That's the important point. The second equation for M, the fraction of active CDC2 kinase, there is a term of dephosphorylation by the phosphatase CDC25 and a term of phosphorylation, which inactivates CDC2, by the kinase V1. And then, where is the coupling? V1 here is supposed to be enhanced by cyclin, upon cyclin binding. And the third equation for the protease, it's, I have difficulties in using my the pointer. So that's a disadvantage of not being present and using a pointer. So dx over dt, it's, so the protease is activated by the kinase CDC2. This is the reason why V3, as in the last line, is shown to be equal to a maximum value times the fraction of active CDC2. If there is no active CDC2, this rate is 0. And if it is fully active, the CDC2 kinase, then the rate is V maximum rate Vm3. So V3 is the phosphorylation rate of the protease, and V4 the dephosphorylation. So how can this system produce oscillations? Because it has negative feedback. Because negative feedback is built in the structure of the system. Cyclin accumulation leads to M activation. This leads to protease activation, which degrades cyclin. So this is the negative feedback. Where do the thresholds come from? Once we will have the thresholds, we will naturally have time delays. So the thresholds come from a phenomenon called zero-order ultrasensitivity, on which I worked previously. So that was unrelated to oscillations. It was a phenomenon on which I worked when I was in Berkeley working with Dan Koshland on a model for bacterial chemotaxis, in which in this model there is reversible methylation of the chemoreceptor. And building a model for that led us to find a phenomenon where you can have very high sensitivity produced by covalent modification of proteins. And we realized that it was more general than methylation, demethylation. For example, it could be applied to phosphorylation, dephosphorylation. It was called zero-order ultrasensitivity because it occurs when the kinase and the phosphatase have a very low Km for their substrate, and therefore are most of the time saturated by their protein substrate. Let us see practically how it applies to this. So I hope I see here, I see here on my screen that I see the room. I hope that you don't see that, right? Because it takes part of the picture. So if we plot the fraction of active kinase CDC2 as a function of cyclin, let us consider only the case A, okay? The case A, that is when there is zero-order draft sensitivity. Then the fraction of active kinase CDC2 varies very steeply as a function of the cyclin level. And this is what happens in the first cycle of phosphorylation, dephosphorylation. In the second cycle, we have the phosphorylation of X, the protease, by the kinase M. So if we plot the fraction of active X, the protease X, as a function of the fraction of active CDC2 kinase, curve A shows that it also displays a very sharp threshold. And so we can discuss, these are steady state curves, but we can use them to discuss what will be, to predict what will be the behavior. And this is actually how the model was built. So if we consider cyclin synthesis, I recall that this is the motor that drives the periodic activation of CDC2 kinase, according to Murray and Kirshner. And we see that C will increase from 0, for example, and then once it reaches the threshold, C star, there will be a large jump in M, okay, from something like, let's say, a few percent to something like more than 90% of active CDC2. Then we can go to the next panel below, B. So you see now that the abscissa has passed from a very low value from the left of the threshold to the right. And the threshold characterizes the activation of the protease X. So when M is increasing sharply in panel A, it produces a sharp increase of X in panel B. And what is the consequence of the activation of X? It will degrade cyclin. Therefore, the abscissa in panel A will go, the cyclin concentration will go from a large value above 1 to a small value to the left of the threshold. And what will happen? As a consequence, M will decrease sharply. Because M, the fraction of active CDC2 will decrease, in the second panel, the abscissa will go from the right of the threshold to the left. M will decrease, and a consequence is that X will decrease sharply. We were on the upper branch, now it will go to the lower branch. What is the consequence of the decrease in X? The balance now between degradation of cyclin and synthesis of cyclin will shift to cyclin synthesis. And once again, we will start to increase in panel A from the left of the threshold to the right until cyclin reaches the value C star, and there is again a large increase in M. Then we go to panel B, we see that there is a large increase in M, followed by a large increase in X, and so on. So this is the mechanism of oscillations. What is remarkable is that the first time the three kinetic equations, which I showed, were integrated numerically, this is the curve which came out. So it's rather rare to have a system of three equations, three differential equations with a number of parameters. Maybe here there are 10 parameters. If you were to look for oscillations by randomly choosing the parameter values, it would take some time to reach an oscillatory behavior. But by discussing the mechanism of the cascade, this is called the cascade of phosphorylation, dephosphorylation, one can predict the conditions in which this will operate periodically, and this is exactly what was verified by integrating the differential equations. You see here that cyclin is increasing linearly at a constant rate. Then it triggers the synthesis, the activation rather, of M, which is the kinase CDC2. The activation of M triggers the activation of the protease X, and this is followed by a decrease in C. The decrease in C triggers a decrease in M, the active CDC2 kinase, and this causes a decrease in X, the protease, and it allows another round of cyclin synthesis. So this is a mechanism for sustained oscillations of CDC2 based on negative feedback. The negative feedback takes the form here of the activation by the kinase CDC2 of a protease which degrades cyclin, which is needed to activate CDC2. Okay. So if you plot that in the phase plane, if you plot the fraction of active CDC2 kinase M as a function of cyclin, this is a projection of the 3-variable trajectory on a 2-variable phase plane, you see that you can reach the same closed curve regardless of initial conditions. And this, so you evolve to the same closed curve, which is called the limit cycle. Yesterday, I discussed limit cycle behavior in a model for circadian oscillations. You see that we recover here the similar type of dynamical behavior. Now, negative feedback is not the only type of feedback which occurs in the system. And in fact, and this is a point on which John Tyson and Bela Novak, who have worked for more than 20 years on modeling the cell cycle in embryonic cells in the frog and also in yeast cells and also in mammalian cells, they stress the role of positive feedback, and rightly so, because, for example, it was shown that the kinase CDC2 activates by phosphorylation the kinase, the phosphatase CDC25, which has a rate V1. Now, remember that CDC25 activates the kinase CDC2 by dephosphorylation, while the kinase V1 inhibits CDC2 by phosphorylating CDC2 on a tyrosine located in the active site of the enzyme. So the fact that CDC2 kinase activates CDC25 amounts to a positive feedback, because CDC2 activates the enzyme CDC25, which activates CDC2. And by the way, there is another feedback where M, the kinase CDC2, inhibits V1. V1 is the kinase which inhibits CDC2. Inhibiting an inhibitor amounts to activating, to self-activation. So there is positive feedback in this system, and you heard another previous talk mentioning the phenomenon of bistability, in which two stable steady states coexist. Very often, bistability, most of the time actually, bistability is based on positive feedback, either direct positive feedback or inhibition of an inhibitor, so which amounts to positive feedback. So the possibility of bistability in the system has been investigated experimentally by two groups. And so both groups published their results, experimental results, in 2003. The group of John Tyson and Gene Siebel at Virginia Tech in the US showed, and this is the result from their paper published in PNAS, showed that if you plot the activity of CDC2 kinase as a function of the cyclin concentration in the abscissa, you progressively increase cyclin, and suddenly there is a jump to a high activity of the kinase CDC2. Then what they did is to diminish the cyclin concentration, and they found that there is a jump to a low value of CDC2 activity, but the jump occurs at a lower cyclin concentration than the jump corresponding to the upper value of CDC2 activity. In other words, this is called hysteresis. Hysteresis denotes this phenomenon where you have a jump between two states which occurs at different limit points. And the curve, the solid and dashed curve which is drawn there, is not the experimental curve. The experimental points are shown, but the curve shows the underlying theoretical curve, where you see that in an interval of two cyclin concentration, you have three steady states. Two steady states are stable, the lower branch and the upper branch, and the middle branch of steady state is unstable. It is shown as a dashed line. So what happens, according to Tyson and also Novak, by the way, it's because they stress that this is a mechanism also occurring in the course of oscillations, is that during oscillations, the hysteresis loop is traveled along repetitively. So cyclin increases, there is a jump in CDC2, which causes, because of the negative feedback I mentioned in the previous model I discussed, this causes a decrease in cyclin, there is a drop in CDC2, and then this allows cyclin increase again, okay? And this has to be contrasted with the model in B, where there is no bistability. And in the model I discussed previously, there was no bistability, by the way, because there was no positive feedback. But one can extend the model I showed by incorporating this positive feedback, and then the results account both for the bistability and for the oscillations. Now, the other group which demonstrated bistability experimentally in the activation of CDC2 is the group of Jim Farrell, who works at Stanford University near San Francisco in Palo Alto. And Farrell wrote a number of very interesting papers, experimental papers, but some theoretical papers as well, on the oscillations of CDC2 in frog embryos. Now, this topic continues to be explored theoretically. For example, this is a paper by the group of Bela Novak, who has worked extensively with John Tyson, and who is a professor in system biology at the University of Oxford. Okay, so as I said, Novak and Tyson produced over the years many models of increasing complexity, both from the embryonic cell cycle. The first reference goes back to 1993. It followed the paper of Tyson in PNAS, which I mentioned before. And the second paper in 2004 is an integrative analysis of the cell cycle control in budding yeast, where they model the yeast cell cycle and account for the behavior of a large number of yeast mutants. Now I will switch to the mammalian cell cycle. The mammalian cell cycle is more complex because we have more than one enzyme CDC2. Actually, as I said, we have 4 phases, in red, M, G1, S, G2. And the transition from one phase to the other, following in the direction of the white arrows, is under control of an enzyme from the family of CDC2. But because it's a family, it was renamed. People now talk already for more than 15 years of cyclin-dependent kinases, CDKs. So there are different CDKs which make complexes with different cyclins. So there is a family of cyclins and a family of cyclin-dependent kinases. For example, the G1 phase is controlled by the cyclin-dependent kinase 4 and 6, CDK4/6, making a complex with cyclin D. The transition from the S phase, in the S phase, the progression is controlled by cyclin E, CDK2. The S to G2, the transition is controlled by cyclin A, CDK2. And then the transition from G2 to M is controlled by cyclin B, CDK1. In the previous model, I was focusing on the situation in frog embryos. The CDC2 corresponds to CDK1, and the cyclin corresponds to cyclin B. So there is also another phase called G0, which is a quiet phase, where the cell sits still, waiting for a growth factor to trigger entry into the cell cycle. So what is the behavior of this network of cyclin-dependent kinases? There are multiple layers of regulation. For example, one type of regulation is exerted on cyclin synthesis and cyclin degradation. Another type of regulation occurs via inhibitors, protein inhibitors of the CDKs. For example, a protein called P21. Another type of regulation is via phosphorylation, dephosphorylation, like in the model I proposed previously. For example, WEE1 is a kinase which inhibits, as previously in the previous model, WEE1 inhibits CDK1, and the phosphatase CDC25 counteracts inhibition by WEE1. So the goal now is to build a model for the network of CDKs driving the mammalian cell cycle. And this is a huge task because of the complexity of the system and the multiple modes of regulation. So I will show that a key role is played here by the level of growth factor, because this is what we will consider as bifurcation parameter or control parameter, if you prefer. This has to be contrasted with the work of Tyson and Novak, who focused on the role of cell mass. And this is rightly so for yeast, for example, where the increase in cell mass is driving the cell division process. In mammalian cells, it seems that it is not as much the cell mass which is driving the process than the growth factor level. So in view of the complexity of the network, it is not astonishing that many works have started by focusing on one particular part of the cell cycle, more specifically the G1S transition. For example, this is a list of papers which focused on the transition between the G1 phase and the DNA replication phase. But when a student, at the time, a student joined me for doing his PhD in Brussels, Claude Gérard, whom you can see here. Actually, Claude Gérard initially had no experience in modeling, and so he started by doing modeling of some simple systems, and then he embarked into modeling the dynamics of the CDK network driving the mammalian cell cycle. And he was not afraid doing so, thanks to his lack of experience. If he had been more experienced, he probably would have been more reluctant to do that. And so there's an advantage of coming to a problem without too much experience in a way, because this does not, I would say, constrain the problem in all its complexity. So what Claude did was to build a model for the CDK network, and because of its complexity, it's necessary to represent it in a manner as clear as possible. And this is already a challenge in itself, because you all know this kind of a picture that is sometimes shown in bioinformatics, where you have hundreds of intermediates, hundreds of arrows and links. And when you see that, my personal impression is that it freezes me, because by its very complexity. So it's important to try to focus on the underlying structure and focus on the key interactions, which of course is by no means easy. So this is the way the model was built. So on the right, you see the cell cycle, which is the succession of the four phases, G1, G1S, SG2, G2, and G2M. You will see why it's decomposed like that, because we considered four modules, four CDK modules. The module cyclin D CDK46 controls G1, cyclin E CDK2 controls G1S, cyclin A CDK2 controls SG2, and cyclin B CDK1 controls G2 and the G2M transition. Now, the important point is that these modules are coupled by multiple modes of regulation. And I will show you the main regulations. By no means this will be an exhaustive model. I will show the model with all the additional regulations after. But I prefer to start with this simplified representation so that you understand what is the wiring structure of the CDK network, the regulatory wiring structure. So we will consider two important factors, which are a transcription factor E2F, which promotes progression in the cell cycle, and a protein which inhibits progression in the cell cycle. And this is the protein PRB, the retinoblastoma protein. Lack of this protein was found to be correlated to certain types of cancer, including one of the retina, hence the name retinoblastoma protein. You will see that the PRB protein can be inactivated by successive phosphorylations. So here we took only two phosphorylations. One, this is intermediate activity, and PRB fully phosphorylated has no activity. And the fully active PRB is non-phosphorylated. Okay. So E2F and PRB are antagonistic factors. And we will see that they play an important role, much as GF, the growth factor. So what is the effect of the growth factor? The growth factor will trigger entry into the cell cycle by promoting cyclin synthesis. Now, E2F also promotes progression in the cell cycle by inducing synthesis of cyclin D, cyclin E, and cyclin A. Okay. Now, this is antagonized by the retinoblastoma protein. It antagonizes the effect of E2F. So what you will see is that these modules, the 4 modules, are coupled in such a way that each module activates the subsequent modules, the next modules, and at the same time inhibits the previous modules. So each module activates the next modules and inhibits the previous modules. And this regulatory structure will allow for temporal self-organization of the whole network. So, for example, cyclin D, CDK4/6, starts to inhibit the retinoblastoma protein by phosphorylating the protein. This will promote progression because PRB inhibits progression. So you are inhibiting an inhibitor. The next module continues to favor progression because CDK2 complex with cyclin E will further inactivate PRB. Then P23 controls P21. P21 is an inhibitor of the CDKs, CDK2 and CDK1, okay? And this here, CDK2 inhibits CDH1, which inhibits SKIP2, which promotes cyclin E degradation. This means that CDK2 promotes cyclin E degradation because it inhibits an inhibitor. So it activates SKIP2. So this is a way that the third module inhibits the second module. The third module promotes the activation of the last module by inhibiting CDH1, which promotes cyclin B synthesis, degradation, okay? So CDK2 inhibits cyclin B degradation. Thereby, it promotes activation of the last module. Now, another negative feedback is provided by the fact that cyclin A CDK2 promotes degradation of E2F. So it stops activation of the modules, the previous modules. And finally, CDK1, the last module, activates CDC20, which promotes cyclin A degradation and cyclin B degradation. So this brings down the activation of the last 2 modules. By the way, the last module is the module I discussed in the model for the embryonic cell cycle, which was based on CDK1 activation of X, the protease. And the protease is called CDC20 here. Okay. So what is the way that this scheme will behave in time? So first of all, what are the types of kinetic equation? By the way, what I show here in panel A is a detailed version of the module number 1, cyclin D, CDK4/6. And you see that it's more complex, but it involves regulation by here P27 and P21, and the effect of E2F and of PRB. So here, for example, one of the equations, the system contains a large number of variables, no less than 39 variables. So one of the variables is E2F. This is the rate of synthesis, the formation and dissociation of a complex between E2F and PRB, formation and dissociation of the complex between E2F and the phosphorylated form of PRB, phosphorylation of E2F, dephosphorylation of E2F, and then degradation of E2F. So because of its complexity, the system contains no less than 39 variables. Now, the model for the embryonic cell cycle contained 3 variables. Now we are dealing with 39 variables. How can we approach such a complex system? Of course, not analytically. There is nothing that can be done analytically on such a complex system. The three-variable model for the embryonic cell cycle can be approached analytically, but not this one. The equations are highly nonlinear, and there are so many. So we have to resort to numerical simulations and bifurcation diagrams. And what I show here is the main result of the model, which is the effect of the growth factor on the one of the variables, which is CDK1, which is let me see. One of the variables is shown here in the ordinate, that's CDK1, as a function of the level of growth factor. Sorry. We see that this is representative of the whole system, by the way. So we have quiescent state corresponding to a stable steady state. And then there is a critical level of growth factor above which the steady state becomes unstable and sustained oscillations occur. Now, this is shown here on the right. On the right, you have two panels. On the top, growth factor equal 0.1. We are here, and there is a stable steady state of CDK1. And this is shown here. There is a flat profile of CDK1. But the next panel, 0.2 for GF, we are here. It's a log scale. We are here, and you see that we are in the region of sustained oscillations. So what is shown here is the maximum of the oscillations that are shown here on the bottom right. We have an all-or-none amplitude. We have the amplitude of the peak here is plotted on the right part. So in dynamical terms, we have a transition from a stable steady state to to oscillations around an unstable steady state. From a qualitative point of view, it is tempting to suggest that the stable steady state corresponds to a quiescent state, no proliferation, and sustained oscillations of CDK1 correspond to successive rounds of cell division. Now, this is shown here. Now we show not one variable, but four variables, the four complexes between cyclins and CDK. And you see that the first peak here is in red. Let's start here. The first peak is cyclin D, CDK4/6. Then the blue curve, which is cyclin E, CDK2. Then the green, cyclin A, CDK2. And then the red, cyclin B, CDK1. And we have successive rounds of activation, transient activation of the CDK cyclin complexes. Moreover, these activations proceed in an ordered manner. Which I just showed before. And these peaks correspond to the phases of the cell cycle, which are represented above. In other words, the regulatory structure of the four modules of cyclin CDKs gives rise to the temporal self-organization of the pathway, of the network, and in the form of sustained oscillations of the cyclin CDK complexes. If now you plot the projection onto a three-variable phase space from 39 to 3. Here we show cyclin B, CDK1, cyclin A, CDK2, and E2F. You see that, once again, the trajectory corresponds to a limit cycle because the red trajectory goes to the same closed curve as the blue trajectory. And one turn on the limit cycle corresponds to one cell cycle. This is the reason why one can say that the cell cycle is a limit cycle, actually. Now, this is the title, which is rather daring, and which covers implementation in the US, Jim York. Jim York worked on chaos, for example. He coined the word chaos. And he likes papers with short titles. And I met him at a conference, and he asked me what I was working on, and I told him that I was working on a paper on the dynamics, on the oscillatory dynamics of the cyclin CDK network driving the mammalian cell cycle. He was horrified by the length of the title, and he asked me to produce a shorter title. And among those I suggested, he said, this is a nice one. So we indeed kept it for this paper. What about, what can we learn from this model? First of all, we can study the bifurcation diagram in a two-parameter space, in a two-parameter space. For example, the rate of synthesis of E2F in the ordinate and the rate of synthesis of PRB. This is a break on the cell cycle. This activates progression. And we have a domain in which we have sustained oscillations and a stable steady state. So the oscillations are observed by integrating the system of 39 differential equations. And so the computer experiment consisted from going from A to B by increasing the rate of synthesis of PRB. Then you should go to a stable steady state. Increasing the rate of synthesis of E2F should bring back the oscillations. Going to D should bring a stable steady state, and back again to oscillations if we further increase E2F. And this exactly is, of course, what was done here. We went from A to B to C to D and E by simply changing the relative values of these rates. So an important result is that what is key here is not the absolute values of the parameters, but the relative values of the parameters related to enhancement of cell cycle progression and inhibition of cell cycle progression. We see this notion of a balance which is emerging. And of course, the balance can be disrupted. For example, if we change the rate of synthesis of PRB and the rate of synthesis of E2F, we see that there is a domain in which we have sustained oscillations which are independent of growth factor, and here which are dependent on growth factor. So this is typical of situations where cells can proliferate in the absence of growth factor. The model can also be used to discuss the existence of our extraction point, but I will try to move a little faster here. What is a restriction point? It is a point beyond which the cell proceeds to finish the cell cycle in the absence of growth factor. And in the model, we can show that if we remove the growth factor after a peak of CDK1, we have to wait a certain time, and after that time, the cell will continue to proceed without growth factor. So this threshold corresponds to the restriction point. What happens in the absence of retinoblastoma protein, if we remove the growth factor in the absence of PRB, we see that oscillations continue. And this can be related to observations showing that when the PRB protein is lacking, then the cell can proliferate nevertheless, which corresponds to immortalization. If we increase P21, the inhibitor of the CDKs, then the system reaches a stable steady state. And it is thought that this corresponds to an arrest in G1, as shown here. It is thought that this is the way which is observed in development. In the course of differentiation, some cells do not differentiate unless there is an increase in P21, which stops the cell cycle. Once again, they can be CDH1 and CDC25. CDC25 is a phosphatase which activates the CDKs. CDH1 promotes cyclin B degradation. I will point this region here, in which this is a region of endoreplication. What is endoreplication? It is shown here in panel D. We have high peaks in CDK2 and small peaks in CDK1, which correspond to successive rounds, successive rounds of DNA replication without cell division. And this is called endoreplication. This, for example, is a review by Bruce Edgar's group, where they focus on endocycles, which are variant cell cycles comprised of DNA synthesis and gap phases, but lacking mitosis. And they may account, they occur often in invertebrate and plant cells. And they are so ubiquitous that they may account for up to half of the world's biomass, which shows that it's an important phenomenon. Now, this network, as I said, is highly complex. On paper, it is possible to isolate sub-networks. Here we isolated 4 sub-networks in the whole cyclin CDK network. And these sub-networks are, each of them is capable of oscillation. But in fact, usually they are synchronized. It's a kind of internal synchronization. If they are not well synchronized together, then one can show that they can give irregular dynamics like chaotic dynamics. It's possible to incorporate in the model checkpoints, because the cell cycle is controlled by checkpoints. And the role of checkpoints is to ensure that the cell does not move to the next phase of the cell cycle as long as the previous phase of the cell cycle is not completed. For example, DNA replication must be completed before so the S phase must be completed before the cell moves to the G2 phase and to the M phase. And so it's possible to incorporate the mechanism of the DNA replication checkpoint, which involves kinases like ATR and CHEK1. And when incorporating these kinases, then the dynamics does not change qualitatively, but you see that the appearance of the oscillations is changing. For example, the green curve is the polymerase, DNA polymerase, which is now an additional variable, and the red curve is cyclin B-CDK1. And in the presence of the checkpoint, there is a better separation between the peaks. This is also shown here. The full system without checkpoint, and in the bottom, the full system with the DNA replication checkpoint. And you see that, for example, cyclin E-CDK2 in blue is more separated from cyclin B-CDK1 than in the absence of checkpoint. Now, is it possible to reduce the complexity of the model for the mammalian cell cycle? This was indeed done in building what we call the skeleton model for the CDK network. Now, the idea behind the skeleton model is to retain the same regulatory structure. So there are still 4 modules of cyclin CDKs, but we remove many biochemical details. For example, there is no phosphorylation, dephosphorylation. It was added later. There is no PRB, role of 4 PRB, and so on, or for inhibitors. And but we retain the same structure, which is based on the idea and on the observation that each module activates the next modules and at the same time inhibits the previous modules. Now, this is much simpler than the full system. This contains 5 variables and 25 parameters. The full system contains 39 variables. You see the degree of complexity. Now, we have now 5 variables. Actually, we have 6 variables, but one is uncoupled. And then this is the result of the study by numerical integration. You see the oscillations in the top panel, and they look very much like those in the bottom panel. One difference is that we do not include cyclin D-CDK4/6 because it does not vary much, and we treat it as a constant in the reduced model. But you see the major result is that you have ordered transient oscillations. No, ordered transient increases, which are repetitive, both in the reduced system and in the full model. We can also include the positive feedback. And as I said, I mentioned already that there is positive feedback. It does not change the number of variables. And but a question which arises is, what's the role of reduced models? Why do we need a reduced model? This is a very nice case to illustrate this issue, because we can use the reduced model, for example, to perform stochastic simulations. Now, stochastic simulations are rather cumbersome to perform, and especially on a very large system, like the 39 variable system. So we use the reduced model to perform stochastic simulations and to address one particular question, which is, why are there so many positive feedback loops in the skin? What's their role? Because they look redundant. So why is it that there is not one positive feedback, but two and even three? So we looked at that by in the skeleton model, and we focus on the positive feedback in G1S. There is one positive feedback because CDK2 activates the phosphatase CDC25, and in G2M there are two positive feedback loops which are already discussed. CDK1 activates CDC25 and inhibits WEE1 by phosphorylation or by dephosphorylation. So we carried the stochastic simulations by decomposing the scheme into elementary reactions and associating probabilities to each reaction. This is based on the work of Gillespie, which is used as a standard method to study the effect of fluctuations. And these are the results. These are oscillations in cyclin E CDK2 in blue, cyclin B CDK1 in red, using stochastic simulations. No positive feedback in panel A, one positive feedback in G1S in B, two positive feedback in C, one in G1S, one in G2M, and three positive feedback in D, one in G1S, two in G2M. What do we see? Oscillations are highly irregular because of noise, of molecular noise, when there is no positive feedback. When you increase in the presence of one positive feedback, you already stabilize, but you stabilize more with two positive feedback and even much more with three. So far from being redundant, in addition to this redundancy role, we see that the positive feedback loops contribute to the enhancement of the robustness of the oscillations. Why? This, by the way, is shown in the phase plane as a projection. We see the limit cycle trajectory, the deterministic limit cycle in red, and the noisy limit cycle in black. So there's no positive feedback there, and you see in the bottom right, in F, there is three positive feedback. And you see that the noisy limit cycle approaches the deterministic limit cycle. Why is it so? Because positive feedback is associated with bistability. Now, I would like to clarify a possible misunderstanding. I speak of bistability, and at the same time I speak of oscillations. They are not contradictory. Simply, bistability is obtained when you isolate the modules and you study each module as a function of its input from the previous module. In such a case, you find bistability. But if you plug all the modules together and allow them to vary in time, then you see oscillations, and the bistability which is hidden controls the waveform of the oscillations. Actually, this is seen here, for example. You see that there is a plateau here in panel D. The plateau is due to the fact that you are going around the hysteresis loop repetitively, and this creates oscillations with a plateau. So what is the effect of positive feedback? It is to enlarge the range of bistability. This is, for example, bistability in the last module, CDK1, versus the previous module, cyclin A CDK2. This is one positive feedback in G2M, one in G2M acting via WEE1, and two positive feedback. And you see that the range of bistability is larger. How comes that it influences the resistance to noise? Because when noise occurs, when the system jumps on the upper branch, it's like a buffer. It does not tend to go back on the lower branch because there is a large bistable range. Here, once you are in the upper branch, you fall down much more easily as a result of fluctuations. So we think that this is one of the reasons for positive feedback. Now, so far, I discussed the role of control parameters located within the CDK network, like CDC25, CDH1. I mentioned the role of GF, the growth factor, which is extracellular. But there are other extracellular factors which control the dynamics of the cell cycle. One of them is the extracellular matrix, because it's known that to proliferate, cells must bind to an extracellular matrix, and this occurs via integrins, which transduce their signaling to a kinase called focal adhesion kinase, and it results in focal adhesion kinase activation, which controls the cell cycle. And also cells contact inhibition, which is affected by cadherins present at the membrane. And cadherins act by activating the Hippo pathway, which leads to inactivation of a transcription factor called YAP, which inside the nucleus elicits cyclin synthesis. So what we did in this paper subsequently is to incorporate, with Gerard, we incorporated the control by contact inhibition and by the extracellular matrix. Previously, we considered growth factors, okay? The extracellular matrix will activate the focal adhesion kinase, which will induce cyclin D synthesis. And contact inhibition will activate the Hippo pathway, which will inhibit YAP, which is a transcription factor inducing cyclin D synthesis. So we have to add several equations describing first the effect of the extracellular matrix, then the effect of contact inhibition, and I will only show you the results. To the right is the CDK1 activation diagram as a function of focal adhesion kinase, which reflects the stiffness of the extracellular matrix. And you see that the bifurcation diagram is similar to the one on the left, which I previously showed, where CDK1 is shown as a function of growth factor. We have first a stable steady state corresponding to quiescence, and then you cross a bifurcation point corresponding to oscillations and proliferation of cells. And to the right, the same occurs with respect to the extracellular matrix. With contact inhibition, as I said, you have a similar behavior because contact inhibition activates Hippo. Now, in the first panel, contact inhibition increases linearly. In the second panel, it increases in a sigmoidal manner. And you see that this leads to activation of Hippo, which inactivates YAP. Okay, YAP is not able anymore to induce cell cycle progression by triggering cyclin synthesis. And as a result, cyclin B CDK1 oscillations in red stop. The same here when you have a sigmoidal increase. So this shows that, to summarize, that there are multiple ways to induce the onset of CDK oscillations. You can overexpress AP1, you can increase skip2. The arrow is the time at which you start to increase skip2 here, and you induce transition from a stable steady state to oscillations. You can decrease CDH1, you can delete P53, you can increase CDC25, and here in the top, in the top you can, you can change E2F. Okay, so, I lost my, here it is. So, to summarize these results, in a graphical way, we can see the cell cycle as existing in two states. One in a cell cycle arrest, corresponding to a stable steady state on the right, one of cell proliferation, corresponding to sustained CDK oscillations on the left. What are the conditions favoring the transition from one to the other? Cell proliferation and CDK oscillations are triggered by high growth factors, high oncogenes, low tumor suppressors, low CDK inhibitors, high EC stiffness, and low sensitivity. And favor the cell cycle arrest. Now, I must add that this view is not shared by all people in the field, particularly by Novak and Tyson, who do not see the transition as being the transition from a stable steady state to an oscillatory state. But I think that one reason is that they are dissecting the network into modules and looking at each module separately. And our approach here, which I illustrated, is a global approach where all the modules are put together and the resulting dynamics is characterized. Now, there is a link to conclude. There is a link between the circadian clock and the cell cycle. And the first link which was discovered is that the circadian clock, which I discussed yesterday with the transcriptional factors BMAL1 clock, induce WEE1, the synthesis of WEE1, which is the kinase which inhibits CDK1, also known as CDC2. Okay, so if we incorporate, and this, by the way, this work was carried out by the group of Okamura now in Kyoto. If we take the model for the circadian clock, which I discussed yesterday, and if we couple it to the model for the cell cycle, which I discussed today, we can incorporate the fact that the rate of synthesis of WEE1 is driven by the circadian clock. There is a parameter for this coupling, which is the rate of synthesis. Okay, and without coupling, if we take a cell cycle of 20 hours, which is shown here, WEE1 is oscillating with 20 hours in the cell cycle, and the circadian clock is oscillating with a period of 24 hours. That's the green curve, BMAL1. They are not oscillating together. In red, we have BCDK1, which oscillates with 20 hours, like the blue curve. If we now make the rate of synthesis of WEE1 dependent on BMAL1, so effectively there is a coupling, then we see here that the cell cycle, which is the blue curve, oscillates, or the red curve, oscillates with the same period of 24 hours as the cell cycle. So this is entrainment. And we studied the entrainment of the cell cycle by the circadian clock by modeling the coupling of these two cellular rhythms. I do not have time to go into all these details. I can come back in the discussion if you wish, but I just would like to show you a counterintuitive result. This one, because it's nice when a model can suggest something which is not intuitive. So we try to entrain the cell cycle by the circadian clock, and we consider two cases. One case where the cell cycle before coupling has a period of 18 hours, and the second case where it has a period of 29.5 hours, longer than the circadian clock. In both cases, we couple via WEE1. WEE1 is an inhibitor. It's a break of the cell cycle. So in the left here, we see that we start here with the arrow by making WEE1 varying in the circadian manner. And we see here that the period goes from 18 hours, the cell cycle period, to the blue curve, which is 24 hours, which is the period of the circadian clock. Okay, this is logical because we put a break and we slow down progression in the cell cycle. So that's how the cell cycle is entrained to oscillate with a longer period, slower. Now, what about the right part? We use the inhibitor of the cell cycle, WEE1, for a cell cycle which has a period of 29 hours. But how can we go from 29 hours to 24 using an inhibitor, using a break? It's counterintuitive because you are slowing down the system and nevertheless it goes from 29 to 24 hours. Now the numerical simulations provide an answer which is quite clear. You see here what happens. We have here the 29.5 hour cell cycle, then we couple it to WEE1, and then this is the resulting period, which goes back, which is now going to 24. What happens is that we narrow, the system narrows the width of the peaks of CDK1. And because it narrows the peaks, now it can be compressed in a way to oscillate with a period of 24 hours. It's like if you put sardines in a can, and then you are really pressuring them so that you can put more within the can. So this is something which is, of course, very clear, but it was not so obvious beforehand how this could be achieved. So I end here, and of course want to stress again the important contribution of Claude Gérard, with whom we continue to work after this work, producing and analyzing this model and publishing the results took 4 years initially. And then, of course, we continued since, because Claude has moved to another laboratory, but we nevertheless continue to collaborate. So I showed first a 3-variable model for the embryonic cell cycle, then a 39-variable model for the mammalian cell cycle, which is a model for the oscillatory dynamics of the CDK network. I extended the model by incorporating checkpoints, the extracellular matrix, and cell contact inhibition. And the reduction of the 39-variable model to a 5-variable skeleton model shows, first of all, allowed stochastic simulations, but it showed that oscillations are similar both in the extended and skeleton models. So a key role is played by the regulatory wiring of the network. Thank you very much for your attention. Sorry to have been long, but a little less than yesterday, and I'm ready to answer questions if there are any.
Any questions?
Let me see. How should I revert to? No, it's yeah, here. Yes. Okay.
Yes. Thank you. Could you elaborate on the difference of using a 5-variable model and the 39 or 40 variable models, like in the sense of which kind of knowledge one model can bring you and the other one?
That's right. For example, I showed a specific example of how to use the reduced model by performing stochastic simulations. This is much more difficult to do in the full model, okay? And this study of the reduced version by stochastic simulations gave interesting answers in regard to the possible role of positive feedback, of the multiplicity of positive feedback loops. Of course, another role of positive feedback loops is to make the transitions from one phase to the other irreversible, as pointed out by Novak and Tyson. But here, we see that another role could be to make the oscillations more robust. So the fact that the oscillations of the CDK networks, or oscillations of CDKs, are similar both in the extended version and in the reduced version shows that what is important is the regulatory wiring. And so we can use the reduced system for studying, for example, populations of cells. That's what we are doing currently. If we did that with the 39-variable system, it would be more complex. But with why do we need then a very complex model, a full model? Because if you ask questions which were specific, like, for example, what is the role of PRB? It's not in the reduced model. So you need to put PRB into the picture to ask what is its role. So the advantage of the full model is that all the parameters correspond to a particular biochemical reaction. And you can thus assess the role of every particular reaction in the dynamics of the whole system. So I think that the two approaches are complementary. And it's interesting to use both, actually, depending on the question you ask.
I was thinking about the hysteresis that you talked about. So it's relatively more straightforward to characterize it while you're modeling using computational tools. I was just wondering how easy or difficult is it to characterize hysteresis in an experimental setup?
Well, first of all, it's more difficult to characterize hysteresis in an experimental setup than oscillations. Because hysteresis means that you have to show the existence of multiple steady states. So it's not so easy to do, and that is the reason why there are not so many examples. That is the reason why the example of CDC2 rather, studied by the group of Tyson and Sibyl and by the group of Farrell, is so nice. But there are two ways to do it. Either you increase the bifurcation parameter, and then you go down at another value. That's one way. Another way is to be located in the region of bistability. And suppose you are on the lower branch, you can perturb the lower branch by adding increasing amounts of the variable plotted in the ordinate. And then there's a threshold. Below the threshold, you remain on the lower branch. Above the threshold, you switch to the upper branch. Thus is a demonstration of bistability. But to demonstrate hysteresis, you need to indeed go back and forth. And it's not so easy to do experimentally. I would say that in a chemical system, it has been done beautifully, but it's easier. What Epstein did on the chemical system is to show that you can increase the rate of influx of a compound, and then you can decrease it. That's when you have a pump. Now with cyclin, it's not so easy to do, of course.
Thank you.
I have two questions. One is, in many embryos, the early oscillations of cell division occur synchronously, then they occur metachronously with a wave flowing down from one pole to the other, and finally the whole thing breaks up and the blastomeres seem to divide out of phase with each other. That's one question. Is there any way in which that can be incorporated in your model? And the second question...
Yeah, first, yes, okay. Now, maybe I can answer this one first.
Okay.
When you can incorporate that into the system by considering space, of course, and a population of cells, and following them in the course of division. So one can anticipate that they are synchronized initially, but then in the course of time, they lose their synchrony. And when they lose their synchrony, each of them becomes a kind of autonomous oscillator. So, but of course, what is the controlling factor that promotes the synchrony and then leads to asynchrony remains to be characterized. Yeah.
What I meant was when they lose their synchrony, there's an intermittent phase during which cell division seems to proceed as a wave from one pole to the other.
Yes.
So there's something else behind it. Okay. The second question had to do...
Then you have to identify what is the controlling parameter, which varies. There is obviously a heterogeneity in the system, maybe a gradient of some factor, but what could it be? So some people have looked at that issue, by the way, theoretically, like in Drosophila, kind of mitotic waves in the embryo. Yeah.
The second question had to do with evolution. Do these models tell us anything or permit us to speculate on how the cell cycle oscillator might have evolved? Does that modular organization that you used give any hints?
Yes. So first of all, this question has been addressed experimentally by people like Nasmith and Nurse, and theoretically also by Novak and Tyson. It's the question arises as to what are the CDKs, the cyclin-dependent kinase, which are needed. Do you need different cyclin-dependent kinase, or can you make the whole system work with only one CDK? In yeast, it has been shown that one CDK is enough, and there is a minimal system which has been built by Coudreuse and Nurse, which has periodic activation of CDC2 with a single CDK. And this has been modeled theoretically as well. But in the mammalian system, it was also shown that although there are different cyclin CDKs, there also CDK1 can substitute for the other CDKs. So there is an overlap of functions. And that was done by Santa Maria in space, in Spain, not in space. And so, so how does it relate to the evolution? Obviously, there is a continuum between the situation in the yeast system and in mammalian cells, where the situation is more complex. But once again, the fact that a single CDK can drive the oscillation is an important element in view of the evolution of the system.
Let's once again thank the speaker. Thank you, sir. We shall break.
Thank you.
We shall break for tea.